Hi everyone, I'm Yitzchak, a master's student under Dr. Nitzan's supervision at the Hebrew University of Jerusalem. Today, I'm going to be talking to you about our work, joined with Noah Moriel, on using co-localization priors and microenvironment statistics to reconstruct tissue organization from single-cell data. First, I'd like to begin with our motivation. In traditional single-cell RNA sequencing experiments, the cells are dissociated from their tissue of origin, and the spatial context of individual cells in the tissue is lost. The RNA expression of these cells is sequenced, which gives us access to the gene by cell expression matrix. Our aim here is to reconstruct the regional spatial organization of the tissue based on this gene expression matrix. Computational reconstruction of tissue structure based on single-cell data has been shown to support the inference of the emergence of structure along development, division of labor mechanisms across tissues, and variation in health and disease. Multiple computational methods have been proposed to approach this task over the past few years, relying on multiple types of prior knowledge. For example, a reference atlas that has information about spatial gene expression of a subset of genes. novoSpaRc is an example of spatial reconstruction method that uses optimal transport to interpolate between prior knowledge about a reference atlas on the one end and a tissue organization principle on the other. Specifically, it relies on an assumption of smoothness in gene expression across the tissue, meaning that cells that are physically close would tend to have, on average, more similar gene expression than cells that are farther apart. While this assumption works well for a variety of simple tissues, like for the Drosophila embryo shown here, for more complex tissues, especially when there isn't a strong enough reference atlas, the reconstruction problem becomes very challenging, both for novoSpaRc and the other available methods. In this work, we show how information about microenvironments across the tissue, what we refer to as co-localization prior, can enhance tissue reconstruction. The co-localization prior here has to do with cell type neighborhoods, specifically the likelihood of different cell types to be located next to each other across tissue. For example, in this image, we can take as a prior that green cells tend to be located next to red cells in the tissue. To do that, we generalize the framework of novoSpaRc to account for co-localization prior. In the paper, we showed that incorporating cell type co-localization priors can enhance spatial tissue reconstruction, and show two examples in the context of the mammalian organ of Corti, a layered structured tissue, and testicular spatial structure, and islet pattern tissue. In the organ of Corti, cell types are organized in layers and spiral from base to apex. And the co-localization prior is based on qualitative description of the organization of the tissue along the medial lateral axis, in which the attraction matrix, the prior, captures the distance between different layers of the cell types in a linear pattern. We mapped all cells together onto a two-dimensional target space. In the left figure, you can see the most dominant cell type inferred in each position when interpolating smoothness assumption and co-localization prior weighted by beta, compared to the right figure using only the smoothness assumption. We found that the two axes of the target space following reconstruction correspond to medial lateral cell type ordering and basal apical ordering of the cells. In the left figure, we can see each cell type distribution projected on the x-axis, and you can see the gradual peaks of the different cell types, in the order of they are expected to be. In the right figure, we can see the apex-based membership projected on y-axis, and the gradual transition between them as we would expect. In the testis data, we computed the co-localization statistics based on cell type annotations of beads in the slide-seq pack. When using only a reference atlas, shown in black with beta equals 0, we recovered for less than third of the beads their actual cell type, even when using large number of marker genes. When we added the co-localization prior by increasing its weight, beta, on behalf of the reference atlas weight alpha, we consistently increased our prediction accuracy. For example, with a weight of 0.4 of the localization prior shown in light blue, we correctly inferred the beat cell type in over 42% of cases. I would like to take this opportunity to thank Moriel, Dr. Nitzan, and the members of our lab. Please read our paper if you'd like to learn more details about our work, and feel free to get in touch. Thank you for listening.